presentation for this session. So Professor DiCarlo is going to give us now his own presentation on basically reverse engineering human visual intelligence. He did some great work on relating cellular net also to the human visual system. So we'll probably get to.
Great. All right, so I'm going to try to talk for 35 minutes. We have time for discussion, because I think that would be the most fun part. I want to thank the organizers for giving me a chance to talk, and it's really been great that Jack and Anitha have set up a lot of what I would have covered, so I'm going to go a little quicker through parts of that. Sorry, can I turn that back down? You guys, is that bothering you? You want to back up?
Yeah.
Okay. How's this? I don't know.
No,
I'd like it better in presentation mode, but if you.
the second
Is that okay?
the one that says spotlight.
Okay. Spotlight. Okay. Okay. I'm sure it's easier for people to see in the other one, but you want this. Okay. All right, so I gave you this big lofty title. Of course, I'm not going to solve that today, but I wanted to try to give a meta part of my talk here, which is to essentially introduce an approach to science that I think is a little different than our field has usually done, but I think is going to be maybe a better way forward for some of these hard problems. And I call that reverse engineering. And what I mean by that is we need to be able to account for each ability of the mind, which I'm calling broadly here intelligence, using, I would say, components of the brain, which for me means neurons and their connections at least. That's the level that many of us most like to think about. It's not the only level. It's what I'll talk about. And in the language of engineering, which means we need models that can be built and tested and distributed to others for building and testing and comparing. That is what I'm calling a reverse engineering approach. Another way of putting this is just do science like engineers. And I'm going to try to show you how we've tried to do that on a problem that we work on called core object recognition. And when I say we, I'm mostly an ambassador for these folks. A lot of the names on the list I'm going to cover work, and I'll try to acknowledge them as they go along. And these are the folks that are currently in my lab, and especially want to acknowledge Danny Gavins, because I'll highlight some of his work. He's now an assistant professor at Stanford. Okay, so, but I'll try to highlight those folks as they come up. So, reverse engineering, as I mentioned, what is that? So my version of this goes like this. So you first have to just define or specify or operationalize a performance domain of interest. And I'll tell you how we do that. So our approach is a bit more top-down than you'll have heard from Jack or Anitha. So we're sort of starting with a behavioral goal. And we are very much motivated by where are behaviors where the systems like humans do better than current engineered systems. This could be better in the performance sense, which I'll show you, but also in the sense of power, size, and so forth. So you're motivated by a phenomenon in instantiation in nature that you're trying to figure out how does this thing actually get that done in such a great way. Then you try to go inside the system and get some measurements that are relevant to how that system might achieve that impressive performance. And here it's an art, really, choosing wisely what you're going to measure, because inside the head there's lots of things you could measure, and including things like behavior, that is muscle movements, and that's not really in the head, but let's say all the other things here, spiking, anatomy, blood flow, neural perturbations, you could do genetics, you could think about synapses and RNA and all that stuff. You could do all that, but you have to choose wisely, and I'll show you our choices. Then, this is a critical step, you have to build something that we'll call models. This is what I'll call forward engineering under the constraints of all the measurements that you have, or at least the ones that you think are relevant to the system performance. And this, for us, means that you need to be able to capture those measurements, and you could call that fitting, so you should at least capture, but you must do more than that. You must predict held-out measurements, not just curve fitting, but actually predict things outside of what you used to constrain the building of that model. Now, for systems neuroscience, this means building models that are called artificial neural networks, because again, those are parts of the brain that we're most interested in here. So that means I'm not happy if models just have probabilities, and we talk about math and probabilities. That may be beautiful if you motivate models, but unless you have neurons in them, I can't map them to the brain, so they're not yet useful to me. It often means that the models must be built at sufficient scale. If they're going to actually predict things in the brain or behavior, they often, these are hard problems, they often have to be built at scale. Vision is a great example. You have 1 million sensors coming in, a 1 million dimensional space coming in. You need to be able to build things that's at least approximating that space. Okay, then you don't just stop there. This isn't just go one way and then you're done. You have to go back and get some more measurements, you do some more modeling, and really it's that loop between measurements and modeling that is the faster you can make that go, that eventually you get something that I'm going to posit we will call understanding. And I think this is a point where we might debate whether that really is understanding, but maybe we can have that debate at the end. And I don't mean understanding of the whole brain or all of intelligence, I mean only over the domain that I defined here as specified domain 1. Now we can expand that domain later, but this is a recipe given a domain. Okay, so this is really science, usually here, measurements, discoveries. This is engineering that is building, but you really need to be doing both of these things together. At MIT, this is represented by the Center for Brains, Minds, and Machines, and more recently, the Intelligence Quest effort, which is essentially a linkage between science and engineering around these kind of issues. And I point out here that if you have these models, they aren't just hoped for applications, they're actually things that should be able to deliver performance similar to the one that you were trying to model or understand here. So you're near to the application. Okay, so I'm going to show you how we've been doing this for really 15 years around the problem of human core visual object perception. That's the domain. So here's how our domain gets started. We really would like to understand this broader domain of visual intelligence, or let's say visual scene understanding. That's a very big problem. So we focused on a subproblem called core recognition, which begins with the idea that you want to label the things out there. As Anitha pointed out, there are other things you may want to do, but for us, we just want to be able to do category and identity. So this is also a problem in computer vision. It kind of looks like this, where you can say these are the objects, these are the bounding boxes. But because we're studying the brain and we know something about the brain, we know it doesn't process that whole image at high acuity. It processes the central region at high acuity, and the part of the brain, the ventral stream, that I will talk about, focuses on the central 10 degrees, which is kind of approximated here. So we're going to think about 10-degree windows, if you will. We also know that primates don't just fixate in one place, but they sample that high acuity sensor, they move it around the scene with saccades and fixations. Those fixations last on the order of 200 to 300 milliseconds in primates. And so what you get is the sampling of the scene at these different points, and that brings to that ventral stream engine a series of images that look like these snapshots here. I'll show you here in a minute. Okay, so those are the samples out of those saccadic points. I'll play it for you again just so you can see. I hope that you can notice that you can recognize and identify one or more objects in each and every one of those glimpses. Even though I kind of pulled it out of context now, you're not driving your eyes around it, you can still do it. And that ability to do that, say 10 degrees in a couple hundred milliseconds, to say there's what the main foreground object is, that's what we refer to as core recognition. And it's not the whole problem, but we think it's the core of the bigger problem. So that's why we call it core recognition. Here's the parameters I just gave you. The task is essentially to report object category. There's other variants to that we've done, but this is the basic thing I want you to think about as the behavior of interest here. So this is all to try to specify something. Why are we specifying this? Well, it's actually computationally very interesting, because even though it seems very easy for you to do that in that short amount of time, it turns out to be quite challenging, and that challenge is due to something that both Jack and Anitha mentioned, which is essentially the invariance problem. The ability to recognize objects in the face of variation, say due to the object's pose relative to you, to background, to illumination, and so forth. So all of that I'm going to lump together as naturalistic variation that the system naturally deals with in a very impressive way. Okay, to test this, we kind of take a middle ground, which is instead of just showing full natural images, we've been generating images that look like this. We generated this because they were challenging for computer vision systems, yet we had control of many of the latent parameters. So these are rendered objects placed on complex, uncorrelated backgrounds, which prevents cheating from the background. Yet I think you can still tell, oh, that's a car, that's a plane, this might look like something from a horror movie. You can still say that's a face or a head. Right? And you can do this even in very short glimpse times, yet machines, again, struggle with this. I'll show you in a minute. Here's an example. This is how a task might actually run for both a human or a monkey. So you fixate this point, and I'm going to flash something up. And then you have to tell me, which thing did you see? I don't know, left or right? said left. Left, right. Okay, I didn't even tell you what you're going to see next, and then this is how we analyze it. We'll try one more just for fun, okay? Okay, so it's to the right. Now, you're pretty good at that. You're not perfect, and we can parameterize that and characterize how good you are, and we've done all that. But here's the big picture I want you to have is here's performance, the prime unit, so this is like 98% correct. This, what we're showing here is that uncertainty, that invariance problem. So this is if you always saw the object right in the center of a certain size, front-facing, and so forth. This is when you have high variation in those parameters, and you can see you're actually very tolerant, not fully invariant, but you're highly tolerant to those variations. Even in the center, 10 degrees. Yet machines, this is around 2009, they were really good at this kind of pattern recognition mode, but not so good at this generalization mode where they have to be able to do this in high variation conditions. And so again, here is the gap that we were especially excited about with regard to core recognition. And you can see, even in these glimpsed images, the system shows its advantage, and that's what we wanted to understand. So I'm talking about this system, so that's why I titled my talk Human Visual Intelligence, because I just did a little test on you. I showed you some human behavior. We really like to understand our own system. That's why we study animal models. But these systems are hard to get that internal measurements for. So we decided to, in my lab, work on this system, which is the rhesus monkey system that we can now say we can get more access to the internal components. Now, the first question you might ask is, well, if I go here, am I going to lose the behavioral performance? And that's one of the advantages of this system is you do not lose the behavioral performance. This is a rhesus monkey doing the same task. This is in its home cage in our housing colony. And you can see the animal's triggering the screen, kind of like you were. When he presses the center, he gets a flash target image. He's choosing among the left and right targets. The objects are all randomly interleaved, so he's not just doing two fixed objects, but he has a whole set of objects that he has to be able to deal with. And green means he's getting it correct, and then he gets a little juice reward. And they love to do thousands of trials like this all day long in their home cage. So we do that, and here are the results of comparing humans and monkeys on these kind of tasks. This is at the object grain. I'll show you the image grain later. Basically, each of these colors indicates the difficulty of discriminating. So you can intuit that tanks are often more confused with trucks. That's this red dot here. And you can see yellow and blue are sort of easier conditions, or easier to discriminate. Now, the point I want you to say here is not to eyeball, how often guns are confused with tables and so forth and so on, but to just look at this and say, which is the monkey and which is the human. And the point is, you can't tell, we can't statistically tell, and at this level, and even at finer grain, these species are indistinguishable in their ability to do these kind of tasks. Okay, so we still have a behavior, now we can go back to go inside and measure the system components. Okay, these are the internals of the system. Jack and Anitha have already introduced it. This is the ventral visual stream. Decades of work have told us that this provides, tells us something about the architecture of the system. It's a series of visual areas in the monkey. Lesions in this part of the brain called inferotemporal cortex that Anitha mentioned produce deficits in recognition tasks, and we were especially interested in that when our lab started. But we're really interested in this whole processing stream. And you can think of it as a sort of a feed-forward, interconnected feed-forward and feedback system shown here. We like to lay out these visual areas and the millions of neurons in each of them in these kind of schematics shown here, which shows their forward connections, their backward connections, and their recurrent connections in these schematic lines. And each dot is meant to reflect a single neuron. Of course, there are millions of neurons in each area. But we know a lot from neurophysiology. Again, decades of work, some of which you heard about already today, but much work that even preceded that tells us things like, hey, we have a map of the visual field at the back of the retina. We have a map all the way up the system. IT, we actually think of as about 3 areas, also as Anitha presented. So you can kind of think of this as 3 regions here, but I'm just showing you for the purposes of today as 1 area. But we also know lots about these neurons, some of which I'll show you a bit later. So this is kind of the rough lay of the land. And then what the idea is you present an image like this one to the system. It activates a photographic sort of isomorphic representation on the back of the retina, and it's rapidly transformed to a new population pattern of neural activity that's schematized here as it finds its way through the system. In about 100 millisecond lag to reach IT cortex. Now, I want you to say here that I'm showing you sort of a feed-forward view, and I've learned that sort of people have decided that DiCarlo thinks the whole brain is feed-forward, and I want to sort of push back on that, say, this is an approximation model to get us started. And as you'll see at the end of the talk, where we're going now is trying to understand these feedback connections. But you've got to get started somewhere. So we work in that feed-forward framework for nearly a decade, and that's the sort of idea of it there. When you have a new image, you get a new pattern of activity, such as this one shown here, and say this rapid serial movie, kind of like those snapshots I was showing you earlier, where you can again recognize one object in each and every one of the images, your homolog of IT clicks along with a new population pattern evoked at every image with a lag of about 100 or 150 milliseconds. And so those patterns are not photographs, but they're very, very special. And I'll show you that in a minute. Here's what it looks like when you record from these neurons in the monkey and IT cortex. And these are now actual brain data. This is, of course, schematic. These are actual brain data. This is just to give you a feel for things. Here's 4 different images shown to a monkey who's just passively fixating. And then these are the different repetitions shown as rows. The dots indicate spikes. This is one neural site in IT cortex. And you see this, like, whatever this image has in it, it drives this cell pretty well and not these cells too well. Here's another site. Here's another site. You can see they're all a little bit different, kind of like you saw from Jack and Anitha. And what I want you to just take from this is we can take this data and we can quantify things. And we do it in a very simple way. Again, simple to get started. And that simple way looks like this. You just count spikes over a window, say of about 100 milliseconds, adjusted for the lag of IT cortex that I already mentioned. And that gives you one average number. So each image for each IT neuron we think of as producing one number to get started. And that number, again, here is an example of computing those numbers. Again, that's not all you want to do, but it's an addition to get started. Okay, that's, as Jack mentioned, you need a lot of data to constrain models. So that was an example, and we scaled up with Utah arrays. I think we were the first to implant these in IT cortex, and these are triple arrays into IT cortex. So we could really scale up our data collection. This was, now about 6 years ago, to get many, many more cells, really more images per cell than we could before using these kind of chronic methods. And when we think about those neural data, then we try to build a linking model, again, back to the behavior of how those neural data actually drive the behavior. And this is older work, but the basic idea is an image evokes a population pattern that we can record. We count spikes in a window, like I mentioned, and I can get one number for each neuron. Now this is a vector across neurons, which you can think of as a point in a neural state space. It's just a way of representing the data. Of course, it must be true because there are neurons that are firing. This is just a description of the data that we observe. So it's sort of a truism that it's a population code. This point here, though, the relationships of these points to each other is relevant when you think about who's reading this information. So these might be images of different faces. Again, this is just schematic here. And images of non-faces, and what you need is a downstream reader to say, I saw faces, to be able to draw a separating plane between those high-dimensional spaces to say, hey, face and non-face, and your car or not car, dog or not dog. So those are the kind of decoders that we build on these neural data, and we think of them as approximations of what neurons can do. Dot products, weighted sums with thresholds downstream. Again, simplifying model goes a long way to explaining what's going on. Here's the actual data that we see. Now this is real data. This is a response to an image. This is typically we collect now somewhere between 100 to 1,000 neurons. Each green color now indicates that average response that I showed you earlier, that I'm showing you again here. So there's a bunch of numbers here. It's a population vector. Here's 8 images, and as I mentioned earlier, we collect thousands of images, so we get data sets that look like this. And I want to point out that these images are collected, these data are collected at very high repetition. So we're really interested in the average spiking response. So we repeat the stimuli many times to get a good estimate of the average response, 50 repetitions or more. So then you have this big data volume here, and a lot of our work we've done so far is to just ask how well this can explain that. What are the linking models between that and that? I introduced that population decode a minute ago, and I won't take you through a lot of work over the last decade that essentially shows simple linear models, those decodes that I briefly showed, can accurately predict these performance patterns in these primate systems going from IT to this. And you cannot do that if you go to even its input area V4, and certainly not for earlier areas. So this is a very powerful set of features here in terms of this linear decodability to support these kind of behaviors. Okay, that's really by way of background. I'll just mention that the specific parameters here are critical and informative to lots of applications, but things like brain-machine interface and so forth, but I'm not going to talk about those here today, those specific parameters. The big picture that I want you to have, though, is that what we know from that previous work is that this representation here is very powerful for supporting the behavior, and we know how to think about it from kind of looking at the spikes as rate codes or rough time windows that we have in mind here, how many neurons approximately, so it gives us a grounding as to what we should be trying to explain back from the image. So we don't just start trying to sort of explain everything that we might see. How are these IT features actually evolved, developed, learned to reach this powerful endpoint? This is one of a key set of open questions. I'm not going to talk about the work we've done on that and are doing on that, but that's one open question that's not answered by what I showed you. And you may wonder, are you really sure that if you push those neurons around directly with, say, optogenetics or use them all, does it cause changes in the animal's behavior in the way you'd predict if you believe this causal linking model that I put up? And we have evidence that that's true. I'm not going to review that for you today. It's not as complete as I would like in terms of able to do full BMI like a matrix kind of thing. But we do have evidence consistent with that. And if we'd like to talk about that at the end, I'll bring it up. What I want to spend the rest of my time talking about is asking, how are the IT features computed from the image? And similarly, what are the internal features? So this is going to connect us to what Anitha and Ed talked about, because they talked about this input area before. They both talked about a lot. Sorry, Ed. Jack, sorry. You guys are basically the same in my head. I know you don't like to hear that. Yeah. Sorry. Sorry. Okay, so everybody with me so far? Let's see how we're doing on time. Let's see. Okay. I've lost track, but we're okay. But I'm going to fly through some more here. So, okay, image computable models. I don't have to say much about this because Anitha and Jack both did, which is you have a model that wants to take an image and have a bunch of feature sets or neural approximations, artificial neurons from that. The models I'm going to tell you about today, you can think of them as working again on the central part of the field, like some roughly 4x4, about 8-degree image. That's how we map these models. So they don't really do the whole task. They're really processing the image to a representation that's one step away from the task. These models are motivated by a lot of neural data that I briefly reviewed and are outlined here. Again, I think Anitha and Jack both outlined some of this as well. This is just to remind you that current deep convolutional nets actually came out of a lot of constraints from visual neuroscience. Fukushima, Anitha mentioned this. The HMAX model that Anitha mentioned, that's like a decade old. And Dave Cox, who's now who was my first graduate student, who's now leading IBM's efforts in Cambridge in this area, and Nicholas Pinta, who's at Apple, built this class of models in the lab. This was in 2009, running GPUs to search these model families. Don't have time to tell you about that, but we were sort of knew that there was going to be good models in this space at the time. But then in 2012, I want to tell you about this model because it was a breakthrough for us. It was the first convolutional network that was able to start to predict these things as well as you saw from Jack and Anitha. And we call that model HMO. The model does not matter anymore because HMO has already been surpassed, as I'll show you. But it was revealing for us at the time. And this was by Dan Yeamans and Ha Hung, a postdoc and graduate student. And here's the gist of this model and what they did. It's a deep convolutional net. Why do we build a deep convolutional net? Because that's what the visual system looks like. It takes filters that applies to different orientations in V1. It applies them at different points in space. That's implemented as a convolution in these models. We don't think it's a convolution in the brain, but a convolution approximates the fact that you're doing that in parallel in the brain. And then you have some nonlinearity, and you do have another set of features in the next layer, and another set of features, and the stack gets deeper, but you're still doing some convolutions over space. You're gradually trading features, space for features, as you see here. The features are built as sort of a linear, nonlinear operator that's motivated off of things that Carandini, Heeger, Mopshin, and others have motivated, and especially for V1, so they're at least biologically inspired, if not perfectly biologically matched. But I like to say that neuroscience put a lot of the constraints on this model family. That's why we were building them, because we're neuroscientists, we're not computer vision people. So then you take those models and you say, get them to do something. This was the main trick that Dan ended up using to really get this to work, is that instead of trying to fit to neural data, which as Jack pointed out is quite limited, you get a lot of data out of the task, or you can get a lot of images and say, if you can guess at the task of what you think the system is doing, it provides a way to tune all the parameters in the model to do well on the task. And again, we chose a task, core recognition, which I've already introduced to you. We can synthesize images as shown here, get it to do core recognition. And then you use basically a bunch of computer science and math tricks to tune the parameters of this model, to optimize them to do well on this task. I don't claim or believe that these are biological. That's an interesting debate. For us, they're just a way to get this network into an end state where it looks like this, and then we can take this thing, which has never seen any neural data, and ask, how well does it look like the brain? So there's no neural fitting here. You just have this network optimized to do something well that kind of looks like the brain architecturally. And now we can ask, how functionally does it look like the brain? So you can take the, quote, neurons. These are neurons in here, they look like planes. The single neurons in here, artificial neurons, compare them with different levels of the brain. And you already saw Anitha and Jack doing this. And sort of a lot of people are doing this now. But again, in 2012, when we did this, this was kind of exciting that we were able to take these things and say, take these neurons out of here, basically treat them as a linear regression onto the neurons here. And the goal was just to predict the responses of the neurons on held out images and see how well we could do that as a measure of match between these models and the actual ventral visual stream. So remember, here's the neural data, kind of I showed you earlier, these are real neural data. We have, of course, the model doing this, generating the same kind of internal data. And then we can take the model and ask, how well can I predict these neural responses here? So here's one of these IT neurons. Here's a bunch of those natural images that I showed you earlier. So there's 1,600 of them here laid out here. This is not time. These are just images plotted here. You might say this is a chair neuron. I don't know, it's a plane neuron. But you can see it doesn't like all images of chairs. There's some kind of structure in the image that it likes that's hard to see through, but you could give it names if you like. But so here's what the model's able to do. This is just the HMO model, taking its layer 4 and just regressing it to predict these images. So the model's never seen these images. It's never seen even these objects in these images. Yet you can see that it can actually generate a very good prediction of the response of this neuron to these images. So that was pretty impressive to us, and that we explained on the order of about 50% of the variance in these responses here, of the explainable variance, which was way better than any model at the time. Here's a face neuron. I heard you guys in the introduction talk about face neurons and so forth. You could call it a face neuron. It would qualify as a face neuron by the standard criteria of face neuron, but you can see it's not really a face neuron because it doesn't like all images of faces. There's structure in here, and you need a model that can actually explain that. But here you go. Here's the model. It can sort of explain all that detailed structure, even though it's hard to put a human name on it, as Jack said. Okay, so again, these models are quite good. They were explaining about half of the explainable variance at the time. So that was IT. Of course, we also applied these to V4. You see a lot of folks doing this now, but here we were applied to V4. This is actually a V4 site, and as Jack pointed out, these are really hard to put labels on. I mean, I can't put a category label on it. It's sort of in no man's land, as Jack pointed out a number of years ago. But here you go, here's the model, and it turns out the middle layer of the model can actually predict quite well if you look at these lines. These levels not so well, this one not so well. Related to what Anitha said, is sort of a Goldilocks zone where the middles of these models actually do quite well. In this case, the model level was layer 3 of HMO, but that was just one particular model. And again, in 2012, we were explaining about half the variance, which was a big jump over previous models at the time. Okay, so these are predictions. I've already said that. These models didn't see any of this neural data, so that's pretty impressive, I think. I also want to say, people sometimes call these models black boxes, but it's not a black box. We can measure everything from it in detail if we like. So it's just a thing that isn't easily describable in human words, but it's a model. Okay, so I want to also highlight, and it's nice that Jack went before me, that there's a lot of nice related fMRI work. Nico Krieger-Skorde, Jack just showed the work from him and Malek, Gardner, Olivia. Others have since taken this and especially applied a lot of papers in human fMRI to doing these same kind of mappings to the ventral visual stream. Okay, there's a summary of the data. Again, my lesson from this, Jack told his paper lesson, is that you submit your paper to NIPS, then you submit your results later to Science, and they say you can't publish in Science because it's already been published in NIPS, even though it's quite exciting. That was a lesson I learned there. NIPS is a real publication, so you can't publish again. These are the results shown here. This is HMO. It fits a layer V4. This is HMO fitting V4. You see the middle layers fit quite well, not so much the upper layer. And HMO fitting IT again quite well. About half the explainable variance explained here. Okay, the meta lesson from all of this that I hope all of you will take from this, probably the most important thing is not about HMO. It's about this slide here. Is that performance on these models is predictive. Performance of these models on a invariant recognition task is predictive of the ability to fit the brain, to fit IT features in this case. You can see that with a bunch of sampled models here, non-tuned parameters here. Here's some sample models early in the literature that I showed you earlier. And what we think we did with HMO is we optimized performance for tasks, and we ended up with a model that was better than these models on these recognition computer vision tasks, was actually then able to predict the variance at a much higher level. So you're just continuing this correlation here. So use this to get that. So this is a computer vision goal, this is a neuroscience goal. And then this leads to a kind of natural, maybe it's sort of scary if you're a neuroscientist, because you say, wait, I don't need to do any work here. I should just sit back and wait around for computer vision to actually produce a copy of the brain. That's sort of the limit case argument here. And that's what it says here. Let's sit back and wait and see what happens. And it was right about this time, in 2012, that AlexNet burst onto the scene as suddenly computer vision folks were suddenly surprised that these models that were neural networks, how do they work so well? It doesn't make any sense that they should work so well, which I found shocking because we were studying the brain the whole time and said these models have to work well. But somebody finally got it to work really well in AlexNet in 2012. And that's AlexNet. Anitha showed you AlexNet. Here's the performance, here's the performance of computer vision on a computer vision challenge called ImageNet as a function of years. And models were doing okay, and then this model came and beat up everybody in 2012. It was the first convolutional neural network to do so well, and then the orange are all convolutional neural networks from here on out. So they sort of took over computer vision around 2013, and of course they're based on the brain loosely, as I described earlier. And now here we go again. I'm going to plot this again for you. This is a newer plot. Computer vision goal, ImageNet performance, neuroscience goal. This is kind of like those blue dots I just showed you. This is the ability to fit IT. Here's a bunch of models. Here's that HMO level that I showed you earlier. Don't worry that these numbers aren't the same. It has to do with how you normalize. I really just want you to show you the relative comparison. And here's AlexNet. That's the model that I just mentioned and Anitha mentioned. And I remember Anitha and I were talking at a conference. I said, Anitha, just use AlexNet. She was asking about HMO. I'm like, don't use HMO. Just use AlexNet. It's already really good. It's publicly available. And it's great to see that she's using that. Because I told her, look, we know that that's actually kind of the best. Because here's what happens in time. So computer vision teams doing what they want, but they're not necessarily getting the brain anymore, right? So there's a bit of a turnover here. And so you can't just sit back. So it's maybe good news if you're a neuroscientist. Say, don't just wait for computer vision. They're optimizing this. They don't care about that. I'm measuring this, but they're just doing that. And so maybe this is like we can't just wait around anymore to let them do. These are all deep nets. They're just not fitting the brain any better than AlexNet, at least on this one measure that I'm showing you here, which is only one of several measures that we have. But they did meet some of our benchmarks. Like here's the behavior I showed you earlier, humans and monkeys. Here's the deep CNN. This was not true for earlier vision models. This was indistinguishable statistically for us at that level of behavioral data. We've actually made a little game out of this. This is our recent NIPS submission paper, so this is not out. Maybe it'll be out at some point. This is a conglomeration of scores that we call brain scores. So it includes IT fits, V4 fits, behavioral fits. And you can see there is this continuing trend, and you don't see a strong of a turnover. There's a little hint of this flattening out here recently. They have advanced, even beyond AlexNet, on a macro sense, if you count behavior, V4 fit. They're on average still advancing in their ability to fit many of the metrics that we are measuring. So we're monitoring this closely while we're also trying to sort of fit things that are still broken. And this is Jonas Cornelius and Martin Shrimp who made this plot here. Okay, so summary here, where are we? So we have this model. We have now several models. All those models near the top of that plot are ANNs that you could take as reasonable models of the ventral visual stream processing, not the learning, as I said earlier, and just for one domain, not all of vision, not even all of object recognition, just core object recognition that I defined for you. That's a lot of progress, I think, but you can see where I'm pointing to where the openings are. What can we do with this, right? So you say, well, is this really understanding? What can we do with this? How am I doing on time? Who's got the time?
A little bit late, 5 more minutes.
5 more minutes, okay. I will do this cool part here because I know Arrow will like it, and then I'll skip the stuff at the end. So, or maybe I can take a vote. All right, I'll just do the cool part, the new part here, because I think it relates to the V4 stuff that Jack and me showed. So this is something completely new that we've been working on for a couple years. I'll just show you a bit of it. One thing you should be able to do with these is to actually control the darn neurons. If you actually have a model of that, you should be able to sort of pick images and drive the neural population into any state you want, in maybe not any state, but possible states. So this is related to image synthesis stuff that you heard from Arrow in the past. You basically take the encoding model, you build the map to the brain, then you invert the process, and Ethan was talking about one method of doing that. There's lots of ways to do that. We do this closer to what Arrow and others have done in the past. We start with a white noise image, and then you just gradient optimize this to get the thing to drive the neurons to whatever state you decide you want to put them in. We call that a controller image. And then you put this back in, you close the loop, so you're still recording from the neurons, you put it back into monkey, and you kind of see, can I drive these neurons actually into the state of the neurons that I'm recording? Can you actually push the neurons where you want them to? And so here was one of our goals we call the stretch, which essentially drive the neuron beyond any response that we saw over the natural space of images. Here's the kind of an example of this attempt. So here's the predicted firing rate from the model. Here's a measured neural firing rate. This is now an example neuron in V4. See this nice correlation here. This is the summary of essentially what I told you earlier. Like, look how good this model is. It can predict really, really well over a space of naturalistic images. So that's kind of what a review of what I showed you earlier for one example neural site. Here is the best image out of this set of images here. Here's the receptive field that we measured independently. Zoom in on it. Maybe Anitha will tell me exactly what it is, the curve or the point thing in here. It's always hard to say, as she and Jack pointed out. But that's the images, so you can look at it. But then we generated these control images, and here you go, boom. They were supposed to drive this thing up here, and they really did drive it beyond anything we had achieved across this large image set here. It's kind of cool. These are done from initial different seeds. You can see they look perceptually similar, but they're not identical. And so this is kind of a sort of neat thing that the model can control the neuron in a way, so this is kind of closing a prediction loop. So I think that's a stronger test of a model than we've had before. Here's some examples of these from other neurons. You can see they're perceptually different. Kind of reminds me of the texture stuff from Arrow's group, but I think that's kind of fun. This is kind of where it is at the moment. But here's the thing I'm actually even more excited about, is like trying not just to drive one neuron up, but to put the whole population in a state. So if you can push one neuron up and all the other neurons to 0. So here's what all the other neurons were doing when we tried to push that one neuron up with this image. That's the target neuron. And you see they're all being driven up a little bit above their baseline 0 here, which is defined as a response to a noise image. But then we could redo the optimization to find another image that tries to control that neuron to put the population in a so-called one-hot state with one neuron, at least recorded neuron, active and the others not active. Try to drive those other guys down. We're not fully successful at this yet, but you can see by using the model, we're clearly making improvements. These are really still early days on this, but this is kind of, I think, a cool direction of testing models to sort of really, and maybe grabbing control of the visual system in very possibly interesting ways. Okay, what else can we do with this? Well, it's not a perfect model, right? It's one model. We can do it to do control tricks and things like that. We can try to do virtual physiology on it, as Anitha said, but that's not the direction we're taking, right? We want to make a better model in the sense it's going to predict things even better. I said we're only about halfway there for IT. We're higher on V4. We've got to make better models. So I'm briefly just going to kind of show you the kind of things we're doing. Just in the interest of time, we're going to flip through. We're testing natural images. We're getting a lot of behavioral data from monkeys and humans. We can look at that data at much higher resolution than I showed you earlier, image by image resolution. You can see these models are not the same. Now when you zoom in, you can see they're broken here. Again, it's not like one image is broken, it's that the whole pattern is broken. And this is, but we're trying to use that data as constraints to make the models better. This is now showing you the models are now clearly not brain-like. Others have shown this in other ways. They're not even as good as the brain when you measure them carefully image by image. That's what's shown here. And just for the kind of people in the room that are really interested in feedback, because I mentioned that earlier, I'll just give you this kind of tidbit here, which is if you compare, this is now I work with a team that I'm on with Jack and I'm Yuri, if you compare how well, what the brain does when it's actually trying to solve, what it's doing when it's dealing with these images where the model here looks like the brain here in terms of behavioral performance. So these are, we call them computer vision solved images. Whereas here's a bunch of images where the system, the monkey, does way better than the model, at least this model, and so we call those computer vision unsolved images. And we're really interested in what is the brain doing to these images that it's not doing with these, because that's where the secret sauce is in some sense relative to this existing deep neural network here. And here's some of these images. You can stare at them all day long. You can do regressions. It's hard to find anything that distinguishes them, occlusion, and so forth. There's a little bit of weight on those things, but not anything obvious. But we do find something that clearly distinguishes them in the brain so far, and that's work on DiCarlo shown here. And the basic idea is we're going to look at those images now, high-performing images only, and we're going to, just to show you what's going on, I'm going to zoom in and show you what the brain is doing in response to these two types of images, the ones that computer vision has effectively solved as well as the brain, and the ones that not as solved as well. And what we do is we play our standard tricks, go into IT, see what the code looks like. You plug in a bunch of arrays down here, you record all the channels, you can then plot the decode as a function of time that I'm going to show you here. So you get this vector, you build the decoder, everything I showed you earlier in the talk. Here you go, here's the decoder. We're now going to plot the decoder as a function of time. Here's one of these images that's solved, it's a face. And I'm going to show you how IT looks temporally now zoomed in, not just one number, but temporally zoomed in. Because we've got much higher resolution now. And what we see here is the time up to the monkey decode. Here comes, boom, about 100 milliseconds. Looks like it gives the animal's report, similar to what I showed you, I suggested earlier. It's this, feed-forward system. Get the answer. Here's a zebra. Boom, comes up. There's the answer. Pops out of IT. You can decode it. Now, here's this interesting image. It's a car. Looks like a pretty easy car. It's a lot to the left, maybe. I don't know, but it's pretty quick to say it's a car. In fact, we selected these images, so you're really good. You're equally good at saying this is a car as you were saying the other one's a zebra. And then here you go. Uh-oh. IT decoded it. It was successful, but it took a little longer. Something happened there, maybe. It took a little longer. Look, IT's responding. That's what's up here. It's not like there's no activity. It's just not linearly available to support the decode. Here comes another one. There's a dog. It's from a foreshortened view. Here comes a little bit longer again. That's 4 examples just to give you a sense. Here's about a dozen examples. And you can see that the red dots, which are the ones that are computer vision not solved, we say, take a little longer to spit out an answer in IT. And this is just one of, again, so many examples. Importantly, these images are all solved in IT. So that's good. Even though they're not solved by computer vision, they just take a little bit longer, about 30 milliseconds longer. So still within a glimpse, still within a couple hundred milliseconds, but something is churning for another 30 milliseconds or so. So all this suggests some role for recurrence or feedback, we think. And this is not maybe surprising to all of you. We just are cool that now we have evidence of where to kind of pinpoint this. And what another piece of evidence here, if you take a deep neural network like the Alex that I showed you here, here's its ability to fit IT responses as a function of time, now zoomed in over time. It fits well at the beginning and then quickly falls off. It doesn't fit so well over here. So all of this is consistent with the idea that these models are missing some recurrence here that exists in the brain that's giving it an extra boost, especially for these kind of red kind of challenge images, we call them here. Okay, so I already said all that. I'm going to just sort of end here and say, okay, you got to build not just a feed-forward network, but we with our collaborators are trying to build networks that are recurrent, that engage that in more interesting ways. That's kind of what that says here. In the broader space, we're excited about the convergence between measurements of the mind and the brain and building things as models, as I've sort of been describing as a reverse engineering approach. We think that's broader than just vision. It extends to other aspects of intelligence. There's my shameless advertising if you're interested in coming to visit us in the lab. And I'm going to just end here with the organizer questions that they ask and my quick answers to them and put them up for discussion. But maybe I'll stop talking in the interest of time because I'm probably going to end. Okay.
Thank you very much. We have some like 3 questions for Jim here, but also for the other speakers till 5 o'clock. So, who would like to?
I have a sort of methods question. You mentioned that you were basically doing the immediate response and not studying learning per se. At the same time, you mentioned that in order to get a good handle on the time course of these responses, you might have played each image 50 times. Can you speak to the role of adaptation and the nervous system when in the real world only sees these images once, and yet you need 50 trials to be able to decode.
Right, so the
adaptation and the real-time problem.
Right, so the form of your question is essentially kind of like a Heisenberg thing. It's like you've got to measure the system and you're going to push the system. So even getting your measurements is moving the system. Right, so
yeah, does it occur?
well, so here, three things I can say about that. It's like if you take data from a passive viewing animal, which is still ends up seeing them 50 times. I don't think we've tried to zoom in on the first image. Others have looked at this. I'm not going to say there's no effect of this, right? There's always adaptation within trials. There's sort of slow adaptation over longer time scales. We're essentially running a little bit roughshod over that because we just average over lots of things. So we're giving you sort of a macro view of the average response over many images. So I'm not going to deny that those effects exist in the data. I can just tell you just a passively viewing monkey versus an actively viewing monkey early in its training versus an actively viewing monkey, late actively viewing monkey early in the data collection versus very late, months later in the data collection. Those 3 look very similar from the metrics used here. So with regard to the ways I process the data here, there's not much effect, but again, it doesn't mean there's no effect. There's also spikes, and again, lots of details in there that the analysis kind of just didn't focus on. So I think I'm sort of avoiding your question, but telling you from the measures we do, they don't seem that dependent on that. But learning is a big open question. I'd say less about adaptation, but again, how do you get the neurons into that state? And I mentioned the animal has to learn the objects from IT. I said there's a linear decoder. The animal has to learn that. He learns that in about 2 days. I can show you slides on that. We don't think that learning is in IT. We think it's past IT. But that's another question I didn't answer for you here, but we have some data on it. But the really deep question is how do you build the ventral stream in the first place? Not probably by backprop with supervised training with a million images, which is kind of how it's generally built.
This is kind of a follow-on to David's question. What percentage of the variance is the explainable variance?
So it depends how you're going to measure the raw variance, right?
Yeah, right.
So if we measure it on, let's say, the count of the count of spikes in that 100 millisecond window on one trial,
Yeah.
that's one number,
Yeah.
right? Versus I can measure on a small bin that's going to be a different number, right? So you can get any number you want. So for us, what's explainable is essentially determined by how much data we collected. Like when we say we have 50 trials and that gives us some amount, 50 reps gives us some amount of explainable variance, have 100 reps, we'll have more explainable variance, right? So it'll just go up. Maybe what you're asking is, well, I'm not sure how to, it's just an open question. I mean, it goes on forever, right? There's no, I don't know, how do you want me to phrase an answer to that?
Within your count windows, you can easily compute the variance just over the trial. You've got 50 trials, so you've got a variance.
Well, so I maybe didn't mention that because everything, we take the data of the 50 trials, split them into 2 halves, correlate them with each other, adjust that for the split half, Spearman Brown correct. That tells us the ceiling limit on what any model should be able to do given that data set. But I could collect a bigger data set where that number would go up, right?
It's always based on repeatability. It's physiology. Something's repeatable is explainable, stuff that's not repeatable is noise.
Yeah, but, no, I understand, but is it really noise?
No, one person's signal is another person's noise.
Right.
And vice versa. So.
And I'll tell you, the really kind of the scary question for us, not scary, but we're trying to address it related to this. What's the explainable variance across animals? That's something that's much harder to quantify in these experiments, and neurophysiology, a small number of monkeys. We can quantify the repeatable variance within a neuron, within an animal. That's kind of what Jack and I and Anitha all do. But cross monkeys is a harder but important question. And it's important to ask when a model's bumping up to a limit, like 50%. That might be the species limit. We don't really know the answer to that question. These guys want to push back on that, but we're trying to figure out what that number actually is, because it's not fair. The model just has to be a member of the species. It has to be like a primate. It doesn't have to be like that particular primate. That's too hard. That's not the model we're going for. That's a different kind of model.
That's legitimate. People have done that in MRI.
Yes, they have many more subjects.
Yeah.
Right.
But also, as apropos of that last point you brought up, there's also the similar statistics issue, which is how much of the explainable variance in your species or in an individual is just due to similar statistics that everybody is processing, versus how much is some sort of unique take on that, some unique riff.
Yeah, I think that's a rephrasing of the same interesting question, right? Yeah. And you could have models that are after the shared component, which is what we're mostly after at the moment. But you also like models that could account for the individual variability, but that's, maybe you're able to engage on that. We're not so much yet.
I wonder how one would embody feedback into this model. For example, there are data from Bar's laboratory in Israel that the low-frequency components can inform IT much faster and then into lower areas. Is your model amenable to such modifications?
Well, yeah, that's where I kind of briefly breezed through here, where I said, look, I'm showing, here's kind of this was kind of evidence that convinced us that kind of timing likely implies feedback dynamics that are important to the animal's behavior, and we have newer decoding models that take advantage of that to connect the things that Mike Chandler talked about. But now you're asking, how do we go from beyond this model to these models, which sort of decide which of these connections we put in and how do we put them in, right? And the way we've been doing that so far is to build models that have them in, so now the recurrent networks optimizing them in the same way that these models were optimized, which is optimized on an image categorization task, which is a harder thing to do. When I say we, this is often, a lot of this work's going on in David Yeaman's lab right now at Stanford, but also Tommy Koja's lab at MIT, and a bit in my group. But that's our approach so far, is to just change the architecture and re-optimize and see if we get things that do higher performance and fit the neurons better. And so far we're getting a little hints of both of those, but it's nothing I'm ready to sort of really say, wow, we made a huge breakthrough. We're not even sure if it's feedback or just recurrence within an area that might be enough. But we're trying to let the data like this drive us there. So that's our approach so far. We have not solved that problem. It's one of the important open questions. Yes?
A small question in between. So between what was said. So what you're doing segmentation of the program.
We're not segmenting anything.
Yeah, because you say that that's the program. You see when you tell the man
Oh, you mean what we're asking him to do?
Yeah.
Oh, I thought you meant what the models do.
Yeah, but are you really asking? Are you really recognize? What's about recognition?
Well,
But this is,
well, so we kind of giving the same task I gave you, which everybody did out of the box. Like, here, tell me what you see. I didn't say segment, but we're building the objects so they're one object placed on the background.
So is it segmentation or is it?
Well, I don't know if it is. All I know is I'll be a behaviorist now. That's my definition of the task. I measure it. That's the data. Any words after that are baggage.
Because in CNS there will be another step up.
Maybe, or the people that do segmentation point out that, hey, these models are actually really good at driving segmentation, right? So I think the interesting debate is about what architecture should you put in, which relates to this, and what task should you optimize? And then the deep hard question is, how does the brain optimize that? Some of that's evolution, and some of that is going to be unsupervised learning, and those are the learning questions. Those are the axes, if Conrad was here, I don't think he's here. I think those are the important axes to be searching over. That's what we're trying to search over.
So this is a really speculative question, and I offered also to the other speakers. Do you think there's a role for using lesions to try to build better models? So in other words, could you train a network that makes mistakes in the same way that monkeys with a V4 or an IT lesion make, or humans for that matter?
Yes, so our approach to that lesion, I like the lesion question because it kind of gets to what we call direct causality tests. And this is sort of our version of this here. It's like if we have these models that are decoding models with a full encoder on the front end, and you lesion different parts of it, you should be able to take that model and sort of say, I'll make a similar lesion on the model as I will to the animal, and I should see similar effects. At the end of the day, all these are tests of model correctness to predict it. At the end of the day, we're converging toward the model. This is back to the understanding thing I said at the beginning. But we've done a bit of lesion work, some of it optical, that's sort of here in face patches here, but we've also been doing new small injections. People mentioned this in some of the earlier talks, like, oh, it's hard to find effects. We get effects across tasks, and so far these effects are consistent with the models that we have so far, but there's an important subtlety that those models didn't have spatial topography. We have to force the neurons to lay out in the brain, which is another kind of cost constraint. It's not a performance constraint, it's some wiring cost constraint. So we have to put that in before we can actually lesion the model. And that's all work that we have that I didn't have time to tell you about, but this is like some of our lesion data, which are sort of micro lesions, irreversible small in IT with like differential pass effects. So I think they're trying to get at, do we have the right model? At the end of the day, it's all about models. And that's a discussion I'd like to have. Maybe these guys should come up here and because I think we're supposed to talk together, right?
That's why I think it's important that Anitha and also Jack come here also, and they can continue asking.
Because I don't know if maybe the three of us share the same view on like what understanding is going to look like with the model. I don't know if you guys are on the same page, but that was. Should we sit?
Sure. I just want to point out in 2011, Shinji Nishimoto's MT paper essentially did lesions. He had this really cool system where he had a lot of model elements that could be switched in and out of the model, and then he basically took different models in and out. Like, do you need rectification? Do you need compressive nonlinearity? And by doing this and systematically comparing all these little elements, circuit elements, you found which circuit elements were critical for MT and which weren't. So you could do that on a larger scale with your whole network if you wanted. There's no reason you couldn't. Whether it will lead something interpretable or not is a separate question.
You'd like to do things that you think you could do experimentally and to say it produces a similar effect.
I think also the thing that you said about an area, we lesioning an area would be different than what Jack was talking about,
Yes.
different aspects, and that probably might produce more interesting or interpretable results than thinking about it as an area, because these networks don't map onto areas.
Like Jim could lesion just, once he gets the feedback working, right, could lesion feedback.
I would like to follow up on the lesion discussion and ask for your opinion on Barry Richmond studies, where he takes out the whole anti-cortex. And not being an electrophysiologist, I would like to know your opinion. My interpretation was that there is very little deficit on primate responses when he takes out large chunks of anti-cortex, unless it is in the presence of clutter. But maybe did I understand those correct? Or
So there's a lot of it. I said at the beginning, we know this area's involved in recognition. Read the IT literature on lesions. There's a lot of studies that find no effects, right? And so then you look at the stimuli and they say, it was a red triangle versus a green square, and there's retraining time. So part of it, if you're not designing the stimuli to force the computations to have to go through the area, the monkey can reroute, right? I mean, we think that to read out of the rest of the monkey, that is the motor system, the downstream stuff, can read out of areas beyond, read probably V1 if it needs to. So the trick to doing this is like trying to keep the animal engaged in the system that you're in and not reroute it. I know there's a nice, I don't know if Mike's still here. There was a nice study from his lab with optical things of like how first they get effects early in time and then went away, right? So, these new small things seem to work pretty well. We have the monkey can route around these. So but when we measure these kind of effects and we compare the size of the lesion we're making relative to the number of neurons, we see that it's the scale you would expect given the amount of neurons involved. And we have some unpublished work on silencing big chunks of IT that brought the animal down near chance on kind of a variant task. So it's not a clean system. It's hard to be careful with the behavior and then do the lesions. The causal experiments like that are really tricky for both of these reasons, just how you're doing the manipulation and how you're controlling the behavior. Folks like Mike or others may be able to even speak better to that, the old stuff with Bill Newsom on microstim. There's a lot of tricks that have to get played to make that work. There's Mike there, so he can probably speak to this better than any of us.
Well, you just said it exactly right, but I'd like to ask another failure question, which is what constitutes evidence against this entire class of models? So, for example, I'm impressed with post-stimulus masking and not seeing gorillas among the ball players on a white team versus the black team and so forth. And, change distraction, which you call it, not change blindness. But these models presumably don't suffer from that. And why isn't that sort of a telltale sign that this is just not how the brain works?
I think it's absolutely right. I think these models don't suffer from those problems, and I don't think these are models of the brain. The way I think about them is, I'm not very imaginative in terms of trying to come up with models for how neurons might build up their properties. And I look at these things and say, can I analyze them and get ideas from these things that will help me think about how these properties might be built up? So I think you're absolutely right. And another way to think about this is to keep making these models fair, because that will help you improve these models. That's another way to think about these models, and also use these models to make predictions about what the responses should be, and then you go and test those. So that's the way I think about it. But maybe Jim thinks of these.
I would not say these are models. I totally agree with everything you just said, and exactly, especially the part about breaking them. But I don't, as you made an important point, that Mike, you can't dismiss the entire family of deep feed-forward networks. If you phrase it that way, you can never win. Just like you can never, falsify Bayesian models at all, in a broader sense, right? But you can falsify particular models. So part of our job is to pin down, like, is it AlexNet? You have to talk about specific models to even, because any function, even a feedback model, is an important thing, right? Any feedback model can be unrolled into a feed-forward model. It just has to be a very deep one. But it's in the family, so it's not falsified. So it's never actually falsifiable unless you pin it down with other constraints.
I think you have to be very careful about feedback, because, correct me if I'm wrong, but I know of no instance where a neuron receiving feedback from a higher area shows any signs of the properties of the neurons that are feeding back upon it. Okay, so feedback to say this is going to help us, we have to remember to satisfy that constraint. And so I don't think it's true. You can unravel them in either direction.
Okay, so, yeah, so I can show you, I'll show you a piece of data. I'm not simultaneously recording from V4 and PFC, but you do find neurons in V4 that show 2 transient peaks in their response. And we think of that, we haven't done the critical experiment for this, but we think the second transient peak comes from feedback. And why do I think that? We've done the exact same experiment in prefrontal cortex, and the second transient peak in V4 looks awfully long, like the first peak. The PFC neurons have only 1 peak, and it looks very similar in response properties to the peak in PFC, and the timing looks like it could be, but it's not. I'm not recording simultaneously. I don't know that these neurons are connected, but this is so I'd be happy to show you that data, see what you think about that.
To your other point, which was that these models are not accounting for interesting psychological phenomena, I would just like to point out the early visual system is the boring part of the brain. In V4, if you look in V4, for how much modulation there is due to attention or any top-down task effects, it's about 15%. You have an animal in a spatial attention task, the modulation of firing rate is about 15%. If you put the animal in a feature-based attention task, the modulation in tuning is about 15%. So these are not enormous effects. You remember Dave Scheinberg's old study from like 20 years ago when he had animals looking for really tiny targets in very cluttered scenes of where it's swallow task. He reported at the anterior pole that essentially attention was affecting 100% of the spike's response, of the neuron's response. If the animal sort of attended to the target and saw the target and was aware of it, you got a big response. If the animal missed the target, even if it was looking right at it or foveating it, you got no response out of the neuron. So I think there's a huge, and there's a lot of data to show that these kinds of top-down effects increase in their magnitude from the back of the head to the front of the head. And where these models, I think in the back of the head, where the attention effects are not that large, are reasonable models.
Some of that, in fact, has to do with the size of the stimulus as well. Right? Those are tiny stimuli, there were lots of them.
Yeah.
And, yeah.
But the 15% effect in V4, that's just a generic line. That's not sizable.
So I just want to continue this theme of the sort of top-down aspects of this, because there's you have these models running, as you're pointing out, on all cylinders, basically, and the case has been made that the reason we need to have attention is that we can't handle this, basically. It's too much of a cost on brain function. So part of the challenge here is that many of the tasks we do, even in the human vision lab, are done with all cylinders. In other words, sparse environments, as you presented, Jim, simple judgments of objects. And that's not what happens in the course of natural behavior. And we haven't really harnessed this in terms of what is the appropriate measure to use to capture what we are getting from any image at any time. And we can't really test these models in addressing the question of how the brain works until we figure out what it is we want to measure. And so that's as much a problem on the psychophysical end as it is a real question on the neural and the modeling end.
I completely agree with the motivation to expand the domain to something bigger than what I showed, certainly what I showed you. I would like it to be argued that way rather than there must be feedback, it must be important, there must be attention, it must be important. If you argue from, here's a new task that's the next task up from core recognition, okay, now I need to deal with eye movements, right? That's sort of the natural thing. Now, what are we going to ask the humans during that to do? Is it a search task? And, things have been done in that domain, but if we could formalize things in that area, and then build models that have to, some people are doing this, right? We pick a problem that we can define, but I agree with you, motivated from some new psychophysical space and do that well.
Which doesn't really exist yet. I mean, people know it's needed, but it's a very hard problem to figure out how to do it.
Thank you for the great talk here. So one question is, when you're talking about neural activity, how much do you care about the cell types? Because I mean, Utah Ray can only taking care of the excitatory neuron signal. There's also almost always there are inhibitory signals also. So when you make decoder or classifier, think that's going to be huge obstacle possibilities, and how do you overcome this, or how do you?
Yeah, at this point, I mean, regardless of Utah arrays or single cells, I mean, we're reporting from neurons and we don't know what types they are. That's going to be the next generation, you know, trying to decide whether these are inhibitory or excitatory neurons, just use some sort of tagging of neurons and types, and then correlating that to the properties of neurons. Those are experiments to be done as the next generation.
I have a rather heretic question, since I'm talking to three people who have devoted their lives in recording and analyzing neurons and neural activity. Do you believe that if we are to map the entire neuronal connectome, we would have solved the problem of how the brain works? In other words, do you believe that neurons are the only processing unit in the brain?
Those are two different questions.
Yeah.
Well,
So when you say connectome, you just mean anatomy, or do you also mean physiology?
both, yeah. So in other words, is in this anatomy and physiology, neurons the only, perhaps, key player in the brain? And I'm referring, for example, to a 2008 science paper where FERA-V1 astrocytes were found to have narrower receptive fields than neurons in other.
That's one of my favorite papers. It's from Jim's copy.
That's one of my favorite papers too. So could, for example, other types of brain cells such as astrocytes contribute to brain function?
Astrocytes have tighter tuning than neurons for orientation because they have a threshold nonlinearity, right? And astrocytes in primate, in mammals, they take up excess neurotransmitter, they essentially repackage it, and they give it back to the presynaptic neuron. And as a side effect, they have little philopodia on the blood vessels. And when an astrocyte detects that it picked up some neurotransmitter, it releases philopodia, and more blood flow goes through. So astrocytes are the whole reason that MRI works. And it's basically just God's gift to MRI that the tuning in astrocytes is actually better than the tuning in neurons. That said,
You're studying astrocytes.
I study astrocytes, exactly. Well, it's something indirectly related to astrocytes, yeah. So, which are indirectly related to the thing we actually care about. That's exactly true. MRI is very indirect. But the point is that astrocyte certainly plays some role in function. For example, when more oxygen and sugar are available in the bloodstream, that actually probably has an effect on the resting potential of the neurons and how energetically efficient they are. But it's a very indirect kind of effect. I mean, you know, most neurophysiologists think that everything you need to worry about is neurons, and astrocytes are part of the metabolic regulatory system.
Well, otherwise they would be called astrophysiologists, right?
Astrophysiologists, I like that. My answer to your question is you've got to be, you've got to ground yourself in something, and I thought most of us want to ground ourselves in some kind of behavior. If you don't ground yourself in that, anything goes, right? Your study synapses, RNA, it's all there, it's all interesting, right? So once you pick some grounding point, which again is a choice, then there's reasonable guesses about like what are the things that are most likely going to be relevant. So is the connectome in all detail going to be needed to satisfactorily explain all vision? I don't think it's going to be needed, but it certainly wouldn't hurt to have it.
I think I'll do astrophysiology after artophysiology.
So, don't you think that this constitutes looking where the spikes are, like the drunk looking where the light is? It's not a question of neurons and spiking, because we have Randy's suggestion of what's in the guts of the neuron may be a locus for memory. We have, I don't know how many of you folks were here a day ago when Dan Margolies presented the subtle effects of changing ionic conductances on spike shape that lead to pattern changes that underlie behavioral changes. And those conductances are conductances, and therefore they're sensitive not only to the intracellular state, but the extracellular ionic state, which may be heavily, as you suggest, conditioned by the glia. So I wouldn't laugh when we talk about glia.
Again, that's why I have a behavior which is adult inference in 200 milliseconds. So that takes out a lot of these sort of like, if I'm studying long-term development, a lot of these other things are relevant in ways I don't yet know. But you pick a problem with a time scale that like spikes are going to be needed to do that, and so you have a better chance of making progress. It's not denying what you're saying, it's just that may be relevant to some other questions that you could say I've tuned my question for my tool in that sense, but it's still an important question.
In the adult brain, most neural communication is through spikes, vast majority. There aren't all that many gap junctions. There are certainly indirect hormonal influences. There are indirect neurotransmitters that act as general neuromodulators. But the vast majority of information transmission through the system, as far as we know, is through spikes. So if you can build a model of spikes, that's a good model.
So, question over here, which is somewhat related to the last few. We've been talking here about how symbols are represented. I want to change that very slightly to ask, how are object categories represented in the brain? And then ask the three of you, given what you've seen and the direction that the modeling is going. The reason to ask a question like that is one imagines that there might be a concise answer to it. Oh, this is how symbols or symbolic representation occur. Okay, so do you envision that there could be a concise answer, a usefully concise answer to the question given from the perspective of this modeling? How are object categories represented?
I can start. So, you probably, I don't know if you know the cat versus dog experiments from Friedman and Miller, 2005.
I'm at the University of Chicago.
That's right, yeah. So, as I was speaking, I realized that. So when they did the experiments in monkeys, they recorded from prefrontal cortex, and they saw a lot of neurons that seem to respond or reflect the category of the stimulus. So, they responded better to dog categories versus cat categories, and they and basically they did a morphing, so that even though like 40% dog and 60% cat were very close to each other, the neurons seemed to represent the category boundary. And when they looked at IT cortex, the responses looked a lot less categorical. So it looked like they were representing more the visual features of the stimulus rather than the category of the stimulus. And so this is probably something that is learned and trained into the neurons in higher stages of processing, and it's probably based on experience, where you start to where the neural responses reflect more the behavioral relevance of the stimulus category as opposed to the visual features themselves. So that's how I think of it. That it's something that gets trained into the network based on behavior.
I'm okay with that, but what you're finding is, collectively, is a lot of detail about how you have to represent the different receptor fields and how that impacts on the performance of the networks, and it's very exciting. It seems to suggest that there is no concise statement about how the networks work.
That's a different issue from, is there a concise representation category?
So in these networks, if you're just talking about the networks, if you look at the fully connected layers, right, you're starting to see categorical sorts of things. So I showed you some examples of bathing cap, swimming cap kinds of things. It doesn't matter precisely what color it is or what shape it is, but as long as it has the features that match a swimming cap, those units are activated strongly by it. So in the fully connected layers, you start to see categorical sorts of things. Perhaps they look more like neurons in prefrontal cortex.
This might be a question, I hate to bring this up, that's better suited at this stage for MRI than for physiology. The direction physiology is going is record everything all the time, and I applaud that. I think that'll be awesome. But right now, you're kind of, you're localized. You have to be localized. And the MRI is bad in almost every way, except you can look at the whole brain. And if you take the MRI data on this issue, there are two really interesting facts. One that, as Russ pointed out earlier, there's a bunch of semantically selective patches of human cortex that surround the retinotopic areas. Sort of the, you end up with these concentric rings of retintopically organized areas that become decreasingly retinotopic. And eventually you end up with a bunch of patches that are fairly selective. Patches selected for faces, patches selected for places, and hundreds of different categories have these semantically selective patches. And then that's in visual cortex. Those patches of stuff, voxels, right, I won't call them neurons, are driven by visual categories. Immediately anterior to those voxels are patches that are semantically selective, but not for visual stuff. They're semantically selective for categories that are brought up in narrative stories. And if you look at the semantic selectivity of patches behind this board, which are visually driven patches in visual occipital cortex, and patches immediately anterior to them that are not driven by visual categories, but driven by stories or language, they're selective for the same semantic categories. So there's a very rich pattern of semantic selectivity in vision, we think, because in the brain generally things that are close together are usually wired together. We think that the visually selective categories feed into, in humans, a language network for representing semantic categories that is amodal and not tied to vision. Now, monkeys are pretty poor conversationalists. They buzz each other, woo, things like that. They don't have a lot of words, but they certainly can do category tasks. So they must have some internal semantic representation. It's not really language, but it's probably amodal and divorced from vision. And I suspect that what happens is these vision networks feed into higher order networks in prefrontal cortex, which are the ones that Anitha was talking about. But those will be category selective. And there's been a lot of work from Miller and other people showing category selectivity in prefrontal cortex.
My version of your question is, this whole thing is going high dimensional to low dimensional. So IT is probably a few hundred dimensions. So you're a 100-dimensional thing. You may buffer a fair amount of that, like sort of release from immediacy. I may buffer a big chunk of that for long periods of time and only draw when you ask me to report, because it's one linear step away from lots of reports. Now, exactly how you do that, we have lots of ideas, but that's an open question still. But you could also, per the cat and dog stuff, when there's very well-learned things, you're going to include them in the buffer too. So those are special dimensions. But it's probably going to be, in my mind, some mixture. It's not going to be like, here's just cats and dogs. It's going to be a buffering of a lot of things, but it's still relatively low dimensional relative to the sensor space that's available for tasks that you could ask me even later. What did you think you saw? And if you didn't tell me ahead of time, I can report on that. So that's a sort of that's how I think about how it's going to look. And I don't know if that's satisfying to you. And then the key questions are like, how many dimensions did you buffer? What were those dimensions? And how do they change with learning and history? And I don't think we fully know the answers. We have hints of the type where to look, but we don't have answers. That's what the thing about axes on the encoding space or on the memory buffer space. Those are, I think, open, interesting questions that are going to, as Jack pointed out, these models now enable, I think, study of. That's what's going to happen.
But we know from psychology that, people probably know about 5,000 to 10,000 categories. So it's a finite.
5,000 categories can still be embedded in a 10-dimensional space, right?
No, I agree. I'm just saying it is far smaller than the sensor space.
Right. Yeah.
Well, that was my point. So I agree with that. And if you take an individual category like dog, it's not. There are 50 different spatially hugely isolated, sort of domains in the brain that represent dog, and every single category is like that. They're represented in a multiplicity of locations, and the expectation is that each one of those locations is representing different aspects of this semantic category. And we know very little about how that actually works at this point, because we don't have enough essentially statistical power in our brain measurements to really get down below the level of kind of generic categories like dog.
This is where we're running late. How running?
I wanted to ask Jim or anybody, is anyone showing these adversarial examples to your core object as a way of driving a wedge between what the net is doing. I mean, if your net is like other people's nets, it's being blown away by these adversarial examples, then what's happening in IT? Is it not fooled?
So that's why I kind of flashed you that part at the end. We didn't show the actual adversarials. For people who don't know, you take an image, you twist a few pixels around, given knowledge of the model, and then you say it still looks like a cat to a human, but the model says it's a gibbon or whatever, right? So you don't even need to do an experiment because you just did one psychophysically to say there's something broken with the model, right? So clearly there's something broken. We took images that we found, that's kind of what I showed you, those computer vision unsolved images, that were like they weren't tweaked to try me, they were just discovered in a set of naturalistic images. And then we looked at IT, and IT handles them just fine, which is what you'd expect, right? So that keeps the linkage between IT and behavior, but shows you that in our mind, the encoding model of IT is wrong, which we already know because it's only explaining half the explainable variance. And then the hard questions are like, how do we build a better model, which is still unknown, and that gets to the feedback things and so forth. So you can show that it's kind of, we've kind of already done that, you know where it's going to go, and I guess that's my view on that one. I don't know, Jack, if you want to add to that.
Okay, I have one final question out of curiosity. So if you show the monkey an object, right, and then the object should move left, right, what if it doesn't move left, right? You show totally different objects. What will the monkey do?
It depends on the training history of the animal. Some animals, if you train the animal to say, you know, if you randomly do a catch trial in which neither of them matches it, the animal might just wait and go to it. But if you do it all the time, then the animal might. It depends also on your reward, right? Like on those trials, do you reward at random, or do you, if it goes to one of those, do you withhold reward? So it depends on what you train the animal to do. Really, wherever gets them the maximum reward.
I was referring more to the fact that since you're recording the before area, you don't need the movement. The movement for you to validate that it's something. And you see in the before, even if it doesn't move left to right, the object.
I didn't show this, but we can see in IT, not before, that you can see a trial by trial. It's essentially runs the choice probability work in the dorsal stream. You can see a trial by trial shift in, okay, it's the same image, but the animal chose one versus the other, and you can see that in the IT population. That's unpublished stuff so far, so I would not take that to the bank yet, but it's what you'd expect, right?
It sounds very interesting. So I guess you heard all these questions. I guess there's a little more to be done. And I think that's great about this field. Like, it's not like vision is becoming docile or everyone's worried like become more. Hopefully not. But this area is growing and it's very exciting. So I'd like to thank the speakers again.